Good boy. Down. Good job, buddy. Good boy. Break. Good boy. Tahoe. Good boy. Down. Down. Good boy. Good boy. Toe. Good. Good boy. Good. Go and stick him on another one.
Place.
Good boy. Toe. Good boy.
Good boy.
Good boy.
Good boy.
Down. Good boy. Sit. Good boy. Break.
Good boy. Good job, buddy. Good boy.
Good job, Tahoe.
All right.
Hi, this is Tahoe. We dropped him off for a 2-week board and train. Before he was here, we had difficulties between the two of us. He wouldn't listen to me as much. He always listened to Brad. He definitely had an issue with distractions. We were walking him and there was a squirrel. He'd run after the squirrel. Definitely didn't have healing down. I think a lot of his, whenever we had him do sit, all had to be with rewards. Anything we did, he had to be fed a treat. And now he's healing. We just got him to sit up on here using this little remote. We were outside. He did heal from a couple 100 yards, and he is doing sit, and he's got his place, and it's pretty amazing. And it took only 2 weeks. He's awesome. Great. Thanks, guys.
using mitosis. So there has to be a better way to organize that DNA. Like that DNA needs to go to the container store and get organized. Well, what actually happens is that DNA can be organized into these units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means in human body cells, in the nuclei, that's the plural of nucleus, you don't say nucleuses, you say nuclei. In the nuclei, there are 46 chromosomes. So basically that means that DNA is organized in chromosomes. It also makes it a lot easier to move over when you're making new cells. Okay, now here's something that's tricky about mitosis that students get confused with. So if you have 46 chromosomes in a cell, you have to duplicate those chromosomes. That basically means you're duplicating DNA, because remember chromosomes are made of DNA and protein. You're duplicating the chromosomes before you divide. You have to do that because if you're going to make an identical cell that has 46, well, you had to duplicate it before splitting. So if you look at our cell cycle video clip, we talk about an interphase, which is a stage that most of the time cells are in. They actually duplicate their DNA. So during that time, they would have duplicated their DNA. So instead of 46 chromosomes, during interphase, they would duplicate and have up to 92 chromosomes. So that when it gets to mitosis, they will be able to split into two cells that each have 46 chromosomes. And that will be identical to the original cell. So let's get right into the mitosis stages. I like to tell students to remember PMAT. It's a little acronym, helps you remember the P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So you remember PMAT. That's the stages in order. So the very first step is prophase. Prophase, because it's the beginning step, the nucleus is still there, and it's going to go away in a little bit, so this is a stage where it's actually still there. And the chromosomes are visible. In fact, we say they're condensing, which means they're kind of thickening and visible. So prophase, very beginning step. The next stage is metaphase, M for metaphase, and I also like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle, waiting there to be pulled apart for the next stage. Next, the A is for anaphase. In anaphase, I like to think of the A is for away. The chromosomes move away. They're moving to the opposite side of the cell. So they're kind of moving to the poles of the cell. Now, one thing to point out, these chromosomes are not just moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the opposite ends. It kind of helps them move along. The last stage, the T, is for telophase. And in telophase, the chromosomes are actually at the complete opposite ends and new nuclei are forming on each side to make these two new cells. And they're starting to surround the chromosomes on both sides. So I like to think of the T is for 2 because in mitosis, you end with 2 cells. And again, remember, they are identical. You can really quiz yourself online with a lot of mitosis pictures. There's tons of mitosis quizzes online to practice being able to look at the cell picture and naming what stage it is. And remember, we really do care about mitosis because without it, we wouldn't understand how cells grow, how they repair damage, and it's really a lot of our cancer research is dependent on understanding mitosis because cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.